Pancreatic cancer is a serious global health challenge, ranking as one of the leading causes of cancer-related deaths worldwide. Pancreatic cancer is a highly lethal malignancy, and in 2020 alone, nearly 500,000 new cases and over 466,000 deaths were recorded worldwide. Pancreatic cancer is often diagnosed at advanced stages due to the lack of early symptoms that it provides, with around 80 to 90% of cases being identified when surgical resection is no longer viable. For these advanced cases, chemotherapy is the primary treatment, typically involving drugs like gemcitabine or capecitabine. However, these therapies face significant limitations due to severe side effects and the rapid development of drug resistance. Given these challenges, there is a critical need for novel therapeutic strategies. The graphical abstract in this paper provides a comprehensive summary of the study. Panel A illustrates the design of the reduction-responsive nanoplatform, showing small interfering RNA encapsulation targeting monoacylglycerol lipase and cannabinoid receptor 2. Panel B shows the accumulation of nanoparticles in tumor tissues when injected into mice, and Panel C and D focus on the RNAi mechanism, showing how each respective target are silenced using the technology and the subsequent effects. One of the targets of the RNAi nanoplatform is monoacylglycerol lipase. It is a lipolytic enzyme that facilitates the breakdown of monoacylglycerol into free fatty acids. In pancreatic cancer cells, this enzymatic activity supports tumor survival and proliferation. The crystal structure reveals a globular protein comprising a central beta sheet in pink surrounded by alpha helixes in light blue. The active site is positioned in a cleft within this fold, housing a catalytic triad that includes serine, histidine, and aspartic acid. Another target of the RNAi nanoplatform is cannabinoid receptor 2. It is a receptor primarily expressed on tumor-associated macrophages within the pancreatic tumor microenvironment. Its activation promotes the M2 macrophage phenotype, which is tumor-supportive. The crystal structure highlights the typical G protein-coupled receptor architecture, with 7 alpha helix transmembrane domains arranged in a bundle. This forms a ligand-binding pocket within the membrane-spanning region. The small interfering RNAs targeting cannabinoid receptor 2 and monoacylglycerol lipase are delivered by a reduction-responsive RNAi nanoplatform. It features a polydisulfide amide polymer core coated with lipid polyethylene glycol to enhance blood circulation and tumor accumulation. The platform co-encapsulates these siRNAs with high encapsulation efficiency of 80%. These spherical nanoparticles are approximately 110 nanometers in size. These nanoparticles are injected into the tumor microenvironment, where they then enter pancreatic cancer cells and tumor-associated macrophages. They are engineered to release siRNA intracellularly in response to the high glutathione concentration, triggering the polydisulfide amide polymer core's reduction-responsive property. The double-stranded siRNA is unwound by the RNA-induced silencing complex machinery, and the guide strand is selectively retained. The guide strand then directs the RNA-induced silencing complex to bind to its complementary sequence on a target mRNA through base pairing. Once bound, the RNA-induced silencing complex's catalytic component, argonaute, cleaves the mRNA at the site of complementarity, leading to mRNA degradation. The silenced monoacylglycerol lipase gene in cancer cells reduces free fatty acid production, starving cancer cells of essential lipids and reducing their ability to grow and metastasize. Simultaneously, silencing of cannabinoid receptor 2 in tumor-associated macrophages leads to a shift in macrophage phenotype from M2, which are tumor-promoting, to M1, which are tumor-suppressing. The repolarized tumor-associated macrophages produce pro-inflammatory cytokines such as TNF-alpha and interleukin-12. This enhances the immune response against cancer cells. By addressing both metabolic and immune pathways, the nanoplatform effectively disrupts tumor progression and strengthens anti-tumor immunity. Now, we will explore some of the key findings of the paper related to the proposed mechanism. The siRNA-loaded nanoparticles exhibited prolonged blood circulation and high tumor accumulation, a significant improvement over naked siRNA, which is rapidly cleared from the bloodstream. This enhanced retention within tumor tissues was attributed to the protective lipid polyethylene glycol outer layer of the nanoparticles. Panel 5A to 5B reflects the results previously described. The first panel shows the blood circulation profile of siRNA-loaded nanoparticles compared to naked siRNA in tumor-bearing mice. It illustrates the longer retention of nanoparticles in the bloodstream, which protects them from rapid clearance by the renal system. The second panel presents fluorescent imaging of tumor tissues and major organs, such as the liver, kidney, and spleen, 24 hours after intravenous injection of siRNA-loaded nanoparticles and naked siRNA. It demonstrates the significantly higher accumulation of nanoparticles in tumor tissues compared to naked siRNA, showcasing the efficacy of the nanoparticle delivery system. In vivo, experiments were also able to confirm that the nanoparticles concurrently silenced monoacylglycerol lipase and cannabinoid receptor 2 in tumor tissues. This results in a marked reduction in free fatty acid production in pancreatic cancer cells and an increase in repolarization of tumor-associated macrophages from M2 phenotype to M1, causing elevated levels of TNF, alpha, and interleukin-12. Panel 5D and 5G reflects results that support monoacylglycerol lipase and cannabinoid receptor silencing, along with reduced free fatty acid production. The first panel shows the expression levels of monoacylglycerol lipase and cannabinoid receptor proteins in tumor tissues, analyzed using Western blot with tubulin as a control. It compares untreated tumors with those treated with different nanoparticle formulations composed of control nanoparticles targeting monoacylglycerol lipase, cannabinoid receptor, or both. The data confirm that nanoparticles targeting monoacylglycerol lipase and cannabinoid receptor concurrently results in significant downregulation of both target proteins compared to single gene silencing or controls. The second panel measures the relative levels of free fatty acids in tumor tissues. Tumors treated with monoacylglycerol lipase nanoparticles show a significant reduction in free fatty acid levels in both single and dual targeting treatments, confirming that this effectively disrupts tumor lipid metabolism. In both xenograft and orthotopic tumor models, mice treated with the dual siRNA nanoparticles displayed a significant reduction in tumor growth with limited size increase over a 24-day period. They also showed high levels of apoptosis and low levels of cell proliferation, confirming the combinatory efficacy of targeting both lipid metabolism and macrophage repolarization. The study also highlighted the nanoplatform's low toxicity, as evidenced by stable body weights combined with normal high hematological and histological parameters in treated mice. This favorable safety profile suggests potential for clinical translation with minimal adverse effects, a critical aspect for cancer therapies. The paper concludes that the reduction-responsive RNA interference nanoplatform effectively reprograms tumor lipid metabolism and repolarizes tumor-associated macrophages, achieving a combinational anti-cancer effect. Furthermore, the low toxicity and efficient gene silencing demonstrated in the study provide a strong foundation for future clinical translation and exploration of similar platforms for complex therapeutic strategies.